you could actually find that. So on a lab practical, the questions would be: name this tissue type, name a location where you find it, give me a function, or there may be a specific cell that's involved or a specific type of protein in there. And then you can be asked for those things as well, right? And so usually when we get up to the connective tissue, which is the second group of tissue, and those guys are grouped into as loose, dense, and vascular. And then we're going to take a look at some muscle tissue. We'll look at 3 different types of muscle tissue: skeletal, smooth, and cardiac. And then last one, what we're going to take a look at are the nervous tissue. So we'll look at we'll look through a cross section of a spinal cord smear in regards to that, and you got to learn about axons and so forth. Okay. So for the lab, what we're going to do is we're going to start out with epithelial tissue, and keep in mind that this is the stuff that's lining you or covering you. All right. So if you think about your skin cells, right, that is many layers of cells, and the very basal layer is attached to the basement membrane. That's the part that's living and growing, and then the upper layers are the stratified layers, and that's the part that is actually is dead. And one of the big things about the epithelial tissue, the squamous ones that we're going to take a look at, is that they're very thin. Okay, so the very first one I'm going to take a look at is squamous. And where you'll find this is you'll find this in areas that require diffusion. So think about your alveoli, right? That's where gas exchange takes place. You have this very thin epithelial tissue there. Right? If it's too thick, it takes much longer for the gases to diffuse across, so you want it as thin as possible. Think about the capillary beds in your blood vessels. Right? You'll have the simple squamous epithelial tissue there. All right. So those are 2 of the big places. Now, when we take a look at this, we're going to look at frog skin. All right? And this is just a dorsal view of that, and so that you can actually see that. When you take a look at this epithelial tissue here. So here is the epithelial tissue for these guys. You can see how, well, you can't see that they're really thin, but what you're looking at the dorsal aspect of it. Okay, and these guys are thin. It allows for diffusion. One of the biggest things about frog skin or amphibian skin is they have the simple squamous epithelial tissue, mainly because amphibians do about 80% of their gas exchange cutaneously. Right? So if you look on on a frog and you look down where the rump is, down by the hind legs, right? One of the things that you'll find down there is they have a specialized area called the seat patch. And they always try to keep their seat patch in some water. And part of the reason for that is so that they can actually gas exchange right across there. So one of the reasons why they want to keep their skin moist is so they can allow for that gas exchange. Because once the skin starts to get dried out, right, that gas exchange, becomes pretty much non-existent. Right, it's not as efficient for these guys. And so they have this nice thin tissue right there. Right? So the other thing here that I want to show you guys, we'll look for on here is on a blood vessel. Now I'll show you generally where. So I know a lot of you guys have already had anatomy already, correct? Yeah. And so this part of the lab, this portion of the lab should be much easier for a whole bunch of you. For some of you guys, it's going to be the very first time you've seen any of this stuff. Right? Now, when you pull a slide, the slide is going to have a name on there. Okay? What that means is that tissue that you're looking at is on there. But there are other things on there. Okay? And so it's go fish. Okay? So here's where having, the nice thing about today is, you know, you can Google. Okay? That's the greatest thing about technology, is that you got right there in your fingertip. You can Google stuff. So you can Google an image so that you know what you're actually looking at. Or you could, some of you guys may have bought, like I know Juan has the photo atlas, right? So you can take a look at the photo atlas, right? And you can make comparisons. Right? So one of the things here, this slide actually says artery, vein, and nerve section. Right? But notice the first thing that I find on here, here's adipose tissue, here's some other connective tissue, here's a nerve, okay? But what I'm looking at right here is this blood vessel. Here's an artery right here, right? And Ernest, how do I know this is an artery, not a vein?
The thickness of the, I forget what it's called, but it's thick because there's a lot more pressure in an artery versus.
Okay, this part right here, right?
Yeah.
Okay. Yeah, that's your. Wait, who else has had anatomy in here? Lupe?
Yeah.
Oh, you're in anatomy.
Yeah, I am.
Okay. And so this is the tunica media, right? This is that smooth muscle.
There we
Right?
go.
So I'm going to go right over here to this layer. And you see this layer right here on the very inside. So if we take a look at a blood vessel. All right. Here's an artery. There are three layers, three generalized layers, okay, in a blood vessel. So you have the internal layer, that's called the tunica interna. The middle layer, right, that's the tunica media, and then the external layer is the tunica externa. Right? The external layer is made up of connective tissue. The media is made up of smooth muscle. And in an artery, right, you have more smooth muscle, right? You have a thicker tunica media, right, than the external in an artery. So here's an artery. The tunica media is greater than the tunica externa in an artery, right? Because exactly what Ernest said, it's under greater pressure. All right? So you get the greater pressure, and then you get the muscle contraction. It squeezes down. It helps propel that blood. The tunica interna. Here's where you're going to find the squamous cells. Okay? Now, because we're in a thicker vessel, thicker artery, right, and we're not all the way down in the capillary beds where blood flow is much slower, right? So blood flow is going to be high in the larger vessels, and then as it gets down to your tissue bed, right, the vessels are going to get thinner. They're going to get smaller in diameter because the pressure isn't as great. Well, we're also going to have a heck of a lot more of them, right? So flow is going to slow down. By slowing down the flow, right, one of the things that you're going to do is you're going to allow for that diffusion to take place. So here's where you want, in the tunica interna, here's where you want that simple squamous epithelial cell, right? So this portion right here is called the lumen. That's where your blood vessels are going to be. That's where your blood vessels are going to be. Okay. All right. So now here's an artery right here. A vein, on the other hand, here is the tunica externa. This would be the tunica media. Okay. And then we would have the tunica interna inside. So on the vein, the tunica externa is greater than the tunica media. because flow is much slower down there. So that's generally what you're taking a look at. So on this portion where the pointer is pointing to, right, here is that tunica interna. So here you can see the nucleus, right? You can see that layer. Notice how thin that layer is. Now, because we're in the larger vessel, okay, when we're not down in, this is not a capillary. Okay? Because when we get down to the capillary bed, we're not going to see this big old thick muscle layer. Okay? We're not going to see that big thick muscle layer on there. So this is a larger artery. But one of the things here, you notice how it's pretty wavy looking right here in this view? So in an artery, what you'll find in the tunica interna is some elastic fibers in there. Okay? And what you're going to see is, because it's going to stretch, you want that rebound. That portion also is referred to as the inner elastic membrane. Right? So for those of you guys that have already had anatomy, right, this should be very familiar to you guys. So the reason why I showed you this artery right off the bat is because I wanted to show you guys specifically where that simple squamous is. I mean, we're looking at it on frog skin, but you can't really relate to that, right? So if we had a cross section through an alveoli, right, through lung tissue, we'll also find that simple squamous on there. Just to kind of show you guys a difference here. There's another little artery. That's not so good looking. Let's see if we can find a vein on here.
This is all adipose.
That's all adipose right there. All right, so right here, the pointer is pointing right in between the artery and the vein. Let me see if we can get both of them in view. You get half and half. All right. All right, so here's an artery. Okay, artery's here, the vein's over here on this side. All right, so here's your picture that you want to photograph. Okay, paparazzi. This is the one that you want because they're both on there for you. Okay, you don't have to go hunting fish. You only have half of each one, but notice that the that's not good to have up there. All right. So, vein. Oh crap.
Just write it on the board.
Right on the board and then put an arrow.
Nah, let's do this. There's your artery. Alright, so this portion right here, there's your tunica externa. So this portion right here is your tunica externa on the artery. That's number right there. That better? You guys see that better? There's the tunica media. There's your tunica media right there. This is your tunica interna. That'll wake you up. All right. And then right here, this will be the lumen. There's the lumen right there. Okay. All right. Not the best view, but is that focused? So notice how the tunica media on the artery is much thicker than the tunica externa on the artery. And then on the vein side, look at the tunica externa, how it's much thicker than the tunica media. All right? So you got an artery on the right, and you got a vein on the left. All right? So that's what we're looking at right there. So on a lab practical, I may ask you, name this vessel. So now you get there's a nice clear view of all that stuff. All right, everybody feel pretty comfortable with that? All right. Now, the next tissue type that we're going to take a look at is simple cuboidal epithelial. So on a lab practical, like when I ask you name this tissue type, you have to write it all. You can't just write cuboidal. Okay? You have to write simple cuboidal epithelial or epithelium. So you got to write it all. So if you've got big bubble writing, write smaller. Okay? So you may have to write it in 2 lines. So this slide that I'm taking a look at right here, it says epithelia, squamous, cuboidal, and columnar. So this tissue has all 3 on there, plus others. All right. So cuboidal, probably the easiest way to remember cuboidal is it's going to look like a little cube. Right? And on the lab practical, I always make sure that I put one on there that looks distinguishable, right? So that you can tell it's a cube. You don't have to question, well, is it really rectangular or not? Right? One good area to look for these are look for little tubules. Right? Down in our kidney tubules is a wonderful place to find these cuboidal cells. So if you look at these cuboidal cells, notice how these guys right here, right? The height, the length of these, right? Pretty similar. You have this nice centrally located nucleus. So if you look at these guys, right? Nucleus takes up a good chunk of it. These guys allow for secretion. They allow for, the reabsorption of substances. So the kidney tubules are a good place, right? And so because they're involved in secretions, right, certain glands are going to have cuboidal cells. Right? So here's your cuboidal cells that you're looking at, and then the basement membrane. See this dark area right here where the pointer's pointing to? Right there. That's the basement membrane right there. So this is the free surface over here. Right? Here's the basement membrane right here for this cell that the pointer's pointing to. All right, everybody feel pretty comfortable with that? All right, so make sure you recognize this. Right? You can name the tissue type, name a location, okay? Give a function. All right, those are the 3 big things that you want to know about for each one of these tissues. Yeah, kidney's fine. You could say glandular cells, glandular tissue, glands. All right, everybody feel pretty comfortable with these? So on the same slide, I just backed it out a little bit. Come over here to this side. So here's a cross section through probably a rat gut, maybe a little bigger. That's going to fit for that. One of the things that you'll see on here are a whole bunch of different types of tissues. Okay, so here's smooth muscle right here. Here's some other dense connective tissue right here. And then when we get over here in this area, here are the cells that we're looking for. These are the columnar cells. And when you look at columnar cells, the thing that you want to remember about columnar cells, they are taller than they are wide. So think of it like a brick standing up on end. All of the nuclei are going to be down by the basement membrane. Find a nice little. See the nuclei nicely there. What I'm looking for is a nice we can see the nucleus all oriented down at the bottom. You're not going to really be able to notice that these are all column shaped, but what you'll notice that the cells are longer than they are wide. Right? So here, right here where that gap is, that's the free surface. And this portion right here, here's where the nuclei are attached to the basement membrane, right here where the pointer is pointing to. Not too bad. Try all right, here are these cells. Let's see. This will work. These guys will work right here. See these? Here's a group of cells right here. All right. The nucleus are these structures right here that the pointer is pointing to. The nucleus in columnar cells are always oriented towards the basement membrane. Right? So because this is forming a little tubule, you'll see all of the nuclei right here. Okay, right through here. Right. So you can see the edge of the cell. These are taller. Right, here's the edge of these cells right here. Right, they're taller than they are wide. So think of it like a brick standing up on end. Right, that's what you're looking at with these cells right here. Right, and so these columnar cells, one of the big areas where you're going to find columnar cells is in your gut, in your intestines. Right? Some of these may be ciliated, some of them may be non-ciliated. The non-ciliated ones you'll find down in your gut. Right? You know, your stomach, part of your gastrointestinal tract on there. Right? You'll find these non-ciliated ones also in salivary glands, certain types of salivary glands. Right? Now, with salivary glands or with certain portions of your intestines, right, they're going to secrete mucus. There is a specialized cell that releases that mucus. This one happens to be stained. Right? See this reddish color stained cell? These are goblet cells. Notice how they kind of look like a perithecium. All right, remember perithecium, the one that was on your lapraxial? All right, that one looks like a little goblet. Right? The function of this goblet cell is to release mucus, is to secrete mucus. Just one. Just one nucleus.
Is that the structure of the whole cell?
That's a portion of the cell.
Like that oval?
Yep, that's a portion of it. The nucleus is right there. There's the nucleus right there. Okay? Alright. Everybody feel pretty comfortable with that one? Alright, so now just to show you guys, you know, a goblet cell that's not stained. See these little clear areas right here? All of those are goblet cells. So here are goblet cells. So right here, you can see the nucleus right down here, right along the basement membrane, right here, right? And then just to the right of the pointer, there is a goblet cell right there. Right? So this one's where the goblet cells are not stained. Right? So make sure you know what the function, that you can identify this specifically as a goblet cell. And you know what its function is. Its function is to secrete mucus. And then if it's ciliated, what you'll see on the very top portion is that you'll see how it'll look a little fuzzy. You'll see actually cilia on there. So the ciliated ones, think of down in the fallopian tubes or the uterine tubes. Okay? Part of your portion. Some of your upper respiratory tract will have these as well. There are some that give this false impression that it looks like it's stratified, test the durability of the camera there, Austin.
It's pretty durable.
So we got some cartilage down below. So here is pseudostratified. if you look at these cells right here, right, and you can follow all of them down, they're all going to attach right down here to the basement membrane. But you'll see that the nucleus on some are lower, some are much higher. They're kind of squished around. These cells, these pseudostratified cells, are found in areas where there's needs to be a little bit more protected. Right? So, part of your bronchial tubes, right? That upper respiratory tract. The epididymis, the vas deferens, are examples of areas that you'll find this. Right? So you need that little extra durability. Your eustachian tube. Right? Where have we heard eustachian tube before? Right? That's the connection between your middle ear cavity and your throat. Right? What is your eustachian tube derived from? You got it. Yeah. Pharyngeal gill cyst, pharyngeal gill pouches. Right? Alright. So they're pseudostratified right there.
Do you know how to spell that?
Pseudo?
No, eustachian.
EUSTAT.
I-A-N. She thinks it's spelled wrong.
Let me, I have to write it. I was always I never did well in spelling bees. That's how you spell it, CH.
Oh, okay.
Okay. Never could spell very well, but definitely couldn't spell very well when I never got past the second or third round of spelling bees. Right. That's how I always ask my wife, "Hey, honey, how do you spell this?" She got her name on the school, the billing thing outside in front of the school when she was in elementary school. I think she won the 4th grade spelling bee or something like that. All right, everybody feel pretty comfortable with that? All right. So then our last one that we're going to take a look at is the stratified. And with stratified, we're going to go to this upper edge. Okay. So you can see this, all this thick layer right up here. Let me go smaller. This thick layer is the dead skin cells right through here. The living stuff is down over here, down into the dermis, but here's that basement membrane. Here's that basal layer. And keep in mind that the cells, the tissue type is named based upon the type of cell that's at the free surface. So for example, if we have kind of this activity over here, right, or we have columnar, we got cuboidal, and then we have squamous, right? This will be named stratified squamous epithelium because of that cell right there.
If you had like a bunch of layers of all squamous, it would just be
It'd be it is stratified squamous. Right? So these are all simple. Simple blank epithelium. This one is stratified because of the layers. Does that make sense to everybody? All right. So that's stratified. All right, you guys want to go run around for 5 minutes? Who's back? people to